and in our skin, and and um, and we have these three main components: desmosome core, linkers, and cytoskeletons. Now, before we talk about tight junctions, we're going to talk about transitory or temporary cell and cell adhesions. So we have been talking about the ones that are in the heart, very strong adhesions, the ones that they are in our skin, and now, but we have other type of junctions that happen only for a second. So we will talk about, for example, some adhesion molecules that are that happen during the migration of the cells. So if you remember the cell lines that they were crawling that you watched the video, so you have some of them that they need to have temporary adhesions to do that. So we have these neural camps, and we have a lot of adhesion molecules related to immune cells. So I will show you these selectins. And you will hear this word a lot, lectin, and sound like pectin, that you will hear a lot of words that they will be very similar. So be careful. In the next class on Monday after reading week, we will talk about pectin. This time we will talk about lectins that they bind uh, to a specific sugar. Okay, so this section of the class, I will finish the topic of adhesion instead of talking about adhesion that are a long-term adhesion. We're going to talk about very short-term adhesion, and we will focus on three molecules. Um, so I told you there were many type of adhesion molecules, and here there are many examples of those. So the one that we talk here, the E coherent. Duncan, do you remember what the E means? E, one word that start with E. One word that start. Okay, let me write it down. Okay, so E coherent is epithelial coherent. Yeah. See, yeah. So every time, so you see the N over there, N cam. So that one is also easy. It's found in the neural system. So it's the neural cell adhesion molecules. This is the one of the few times that they make life easier for us. Yeah. That so for example here, this selectin, it will be also related to the function. Yeah. So they make it much easier for us. Okay. So we have ecoherent, we have NCAM, we have the selectins, we have integrins. I don't think any of you know about integrins. Paige, you might remember something from summer, but you might not remember anything that from cancer biology. Any of you know anything about integrins? Integrin? Yeah, I know is that this course in cell biology is like take 2,000 new words. Go and remember and forget it in 2 weeks. So integrin is a very important protein that is found between the cells and the cell and the extracellular membrane. So every time that you see a paper on cancer, how they migrate, you will find the integrin. Yeah, we're not going to talk a lot about that in this course, but this is another adhesion molecule. So remember how the cells escape? They go down to the basal membrane, and how they do it using these integrins. Okay, base. I know too many things, too many molecules, and in that cancer biology is even more courses, and every year is more papers and more molecules that they appear. Okay, so I just told you that besides the coherence, there are molecules that are involved in cell adhesion and cell recognition, and here there are some examples. And so we have coherence, yeah. We have this neural cell adhesion molecule, and so just sorry, Duncan. Yes, I know that will be in the test. So if you remember E from epithelial, N from neural, so it will be easier to answer some of the questions. This is these intercellular adhesion molecules. They are related to the immune system. Okay, you will see a lot of those one in a second. Uh, So there are different types. I already talked about integrins. I'm not going to ask you a lot about integrins, about how many repetitions they have, how the transmembrane domain, but you need to know that they are located between the extracellular membrane and the cell, okay? Yeah, that could be one question. Coherence, I will ask you in the test, for sure. You remember that Paige and Derek last time they told that they have they need calcium. They are calcium dependent molecules. If there is no calcium, they don't work. And as I told you, you will have a lot of this temporary adhesion. You have in many antibodies like in immunoglobulin superfamily. So what they need to adhere, they need to attach to a bacteria, to a virus to go and destroy it. And you'll have selectins that we will talk in a second about. Okay, so these are a diagram of all of them. We talk about coherence, we talk about the desmosomes. Here is the integrin, and here you see that um, there is there is not another cell here. There is not, and they are not attached to something else. And here you don't see another one because I will show you why the selectins you don't see another type. Okay, so in 1 second. Okay, so remember lectins. So this one will be start with the L. Uh, it will be one of these adhesion molecules, and here you see the lectin in green. Yeah, and it's in a bacteria, and here there's an epithelium, and these lectins are very good at attaching on specific proteins. Yeah, so and specific carbohydrates. Yeah, this is what lectins are doing, attaching to carbohydrate. And in this case, what they're doing is attaching to those carbohydrates to disassemble the attachment of the cell and attack a cell. Yeah, so here we see a bacteria that is attacking using the lectins. Lectin can be found in bacteria, in plants, and in animals, and they is used in different ways. So what I told you, here I gave you an example, but the idea is that a lectin is a protein or a glycoprotein that especially bind to carbohydrates with high affinity. Uh, and remember, lectin promotes cell adhesion, binding to sugars in the outer cell surface. And the function that they do is they bind to carbohydrates, and three types of those functions. They could be attaching to carbohydrates for cell recognition, for the immune system role, or for pathogen binding, like I show you here. Okay, so lectins very important, one type of cell adhesion. And here I just show you one that is the pathogen binding, very important. But there are many other ones. They could be involved in apoptosis, cancer biomarkers. There are specific arrays that involve lectin and autophagy. So there is a lot of functions that the lectins can be used. Okay, so Asher, uh, super easy question. Can you help me with the answer here? B. Yeah, so that will be pretty easy. The cell adhesion molecules are involved in attaching to another ones. So here you find an example of a CAM. Here is one very famous immunoglobulin. Yeah, so you can see that has a transmembrane domain this in the tail in the cytosol. And here you see that immunoglobulins are commonly known as antibody, and they are produced in the immune system, and they identify and neutralize a foreign substance. So in the previous example, I show you how the pathogens can attack the cell using adhesion molecules. Here I show you the opposite, how the body use these adhesion molecules to actually attach to a virus or a bacteria. Okay, so important. So here we have the cell adhesion molecules. Uh, I told you also that the cell adhesion molecules could be for crawling, yeah. yeah, for migrating. So if you see in the crawling video that you watch, you see that they have to form actin and they start moving in this direction, and they need temporary adhesion to the substrate. So here you have, for example, this neural cell adhesion molecule that here you can see that is homotypic. So this one is the same that the other one, okay? And here you see that they have these well-organized loops. This is not the, later we will talk about our type of loops that they are found in the tight junctions. Okay, so I have told you so far that there are important proteins junctions that they can produce just temporary adhesion. Yeah, they adhere to something in a temporary way, and I show you an example that it was the lectins. I show you the end caps, and now this one that are very important that are the selectins, and here you have the one that is related to the leukocyte adhesion, and I put a video there, and I put the video also in the on Populi, okay, that you want to study later, because I find that it's much easier, and it summarizes that video, summarizes the entire class. So here you see that this one is called L-selectin, so it's found in the leukocytes. The L come from the leukocytes. So Duncan, this E-selectin unfortunately doesn't come from epithelium. This one comes from endothelium selectin. So here you will find the endothelial, and this one will attach to other molecules that I will show you in the video. And here you have another type that is the P-selectin. So remember, they are helping you all the time with those things. If you know leukocytes, they will have L selection. The endothelial will have this E selection. Okay, selectins. And I will show you in a video when a leukocyte is going to go to attack a bacteria to defend the body. You need to go and travel and roll and stop and use this. Like when you play volleyball, these are like the hands of the volleyball you will see in the video. Yeah, so they have to stop it, and it will roll, and there will be other proteins that stop the leukocyte, so it go down. I know it sounds crazy, but you will see it in a second. It's amazing, and I was going to show you a video how the leukocytes are able to go through the membranes. It's amazing. But let me show you just the adhesion. So here you have the leukocyte that they interact with the lining of the vessels, and this is mediated by selectins. You have these L-selectins, remember, for the leukocytes, the P-selectin for this type of cells. Could you help me, Paige? I forgot how to pronounce that. Platelets. Yes, I was going to say plaquetas, and I was like, this is not a word in English. Platelets. Yes, thank you. So here, the P from here is from here, the platelets, and the E-selectin, in this case, is from the endothelium. Okay, so here you will see, again, the leukocyte, that is the immune cell. Here you see the endothelia, and here you see the surrounding tissue. And we are going to see how it's able to, using these temporary adhesion molecules, to stop the leukocytes to go and attack, protect the body.
Leukocytes, or white blood cells, normally circulate in the blood unattached. The presence of an infection or inflammation triggers leukocytes to move into tissue toward the foreign invader in a process called extravasation. Extravasation requires a succession of specific cell-cell contacts between leukocytes and endothelial cells lining the blood vessel. First, inflammatory signals induce endothelial cells to exocytose P-selectin and synthesize platelet activating factor, PAF, both of which appear on the cell surface. P-selectin molecules bind oligosaccharide groups at the leukocyte cell surface. Since this adhesion is weak, the leukocyte rolls along the endothelial cells. The leukocyte also contains PAF receptors and integrins. Receptor binding to PAF results in the activation of the integrins. The activated integrins are able to bind ICAM molecules constitutively expressed on the surface of endothelial cells. This causes the leukocyte to stop rolling and to tightly adhere to the endothelium. The adhered leukocyte then begins its migration between adjacent endothelial cells to reach the underlying tissue.
Okay, so here this is why it was like a summary of all the transitory adhesion molecules. You saw that they were talking about integrins. You saw they were talking about other adhesion molecules. I think they talk about the ICAM. Yeah, all of them, they have to, as they are those cells, the leukocytes are not together to any to other cells. They are just floated in the blood. They need to be stopped and they need to attach. And how they can do this amazing thing? To imagine they have to take the nucleus and make it very thin to go between cells. That is amazing how they do that, and they have to dismantle the adhesion molecules and to get into the other tissue. This is not a surprise for you. Okay, one disease that is able to do this. What disease is able to do this process of extravasation, I think it's called, to go from the blood vessels into another part of the body. So there's intravasation and extravasation. And of course, all the answer here will be cancer. Cancer is able to do that. So they go from the epithelium, get into a tissue, yeah, and then they said we need food, so we need blood supply. Then they are able to, when they get into the blood, it's like taking a highway that they can go everywhere with a lot of food. And then they said, now we need to go to invade another tissue, and they do that. How they do it? Because we have the same genes, so we have all our cells have p-selectins, all of our cells have all the genes to the integrin, so they can do that. So they just need to express it. Is this going to happen in one mutation? No, it will take years and years of that. Okay, so the video, I put it there. So, and I will put it in the practice questions, some questions of this. Okay, and if you want, in the textbook, again, they are telling you 3 steps. There is the rolling leukocytes is mediated by p-selectin. Primarily in the blood vessels, they have integrins. So they are showing you here the integrins here, integrin here, and then allow to the attachment and the invasion. Okay, so here you have the endothelium, and here the leukocytes, as you're watching the video, are able to attach to all those molecules to go inside and go to the other tissue. Okay, so with this, we finish. Okay, first, Angus, hopefully this is easy. No, it will be super easy. What is the answer of this super easy question? A. A. I might be wrong, but this is 2 carbohydrates, so maybe I made a mistake, but this will be lectin. Lectin, yeah? And this is selectin. Maybe I made a mistake, yes? The addition molecules to bind to carbohydrate. Yes, let me see. Oh, yeah, I think that one was lectins, and the selectin is that one that attach to the immune cell. But let me check. Yeah, in the quiz, it appeared like A.
In the quiz, that was the answer.
Okay, so I made a mistake and I have to remove. Thank you. I will move not selectin but lectins. Okay, so sorry for the typo. So lectins are the ones that attach to specific carbohydrates. Yeah. So the answer there should be selectin. For the quiz, all of you will have it right. For the case of for the test, I will fix it. I show you all examples of that it was these neural camps that are homeotypic interaction, and here these selectins that are the ones that mediate adhesion of white blood cells and endothelial cells. Okay,